Good morning, everyone, and welcome to this week's Department of Medicine Grand Rounds. I'm Dana Bowen, one of the chief residents here at University Hospital. Before we get started, I just wanted to share a couple of housekeeping things. Please make sure your video and mics are off during the presentation. We ask also that you hold your questions until the end, at which time you may use the comment box or unmute yourselves, turn your videos on. Lastly, the CME information will be displayed during the presentation, and I'll also put it in at the end of the presentation in the chat box. Today, our speaker is Dr. Mark Siracusa, joining us from the Department of Medicine's Center for Immunity and Inflammation for a presentation on mast cell-mediated inflammation. Dr. Mark Siracusa is Associate Professor of the Department of Medicine's Chancellor Scholar and Director of the Research Support Corps. He earned his PhD in molecular microbiology and immunology at Johns Hopkins Bloomberg School of Public Health, then went on to do research in mucosal immunology at the University of Pennsylvania. During his tenure at Rutgers, he has employed diverse marine model systems and pioneered translational studies to dissect innate immune responses at mucosal sites. Dr. Siracusa has also served as principal investigator of multiple NIH grant-funded projects in the areas of mast cell biology and published dozens of papers in clinical allergy and immunology. He's also the recipient of many awards and honors, including the 2020 New Jersey Health Foundation Excellence in Research Award. We welcome you and thank you, Dr. Siracusa, for joining us today to give this presentation.
Great. Yeah, thanks for the nice introduction and also the invitation. It is a pleasure to be able to present. Hopefully I can do it more often. So today I'll be talking about some of the work we're doing advancing therapies to treat mast cell-mediated inflammation over in the Cancer Center. Just a couple disclosures, then I'll put the CME information up, but it's important that I note that I am the founder and president of a Rutgers startup company named Nemogen Discoveries. A lot of the data you're going to be seeing today is part of patent applications that have been granted to Rutgers, and then we've subsequently licensed them to Nemogen. So I don't receive any financial compensation from this at all, but it's possible in the future I could. Really, this startup company was founded to help with our drug discovery programs, things that are difficult to fund via NIH funding. Most of this work is supported by funds that have been provided by the venture capital group, the New Jersey Health Foundation. I also have to disclose that my wife and I hold substantial financial holdings in Novartis. Nothing to do with my research program. My wife works in marketing for Novartis, not even a scientist, so it's unrelated, but it's reached the disclosure level. So with that stated, this is the CME information. I'll also have this projected as the final slide for your reference at the end, so you'll have plenty of time to take it down. Okay, so what I actually study in my lab is a little bit different than what most people study. Is we actually study parasitic worm infections or Helminth infections. So I'm one of probably very few labs in the United States that study this anymore. So just to give you some background on these Helminth infections, these parasitic worms, they actually infect about a third of the world's population. That's not often studied. These infections are not often studied in the United States because we're virtually void of these infections in the United States as well as the EU. So they infect developing areas of the world, such as South America and nations in Africa. So these are very clinically important infections. They have synergistic effects with other neglected diseases such as HIV, malaria, and tuberculosis. So they don't often kill people, but they cause a tremendous amount of morbidity, and they also drive economic problems in these already relatively poor areas of the world. And then when you start to factor on that they actually have negative effects with HIV and malaria and tuberculosis, those infections are also highly prevalent in the same areas that Helminth and parasitic worms are present. One of the other issues we're having, I just sat on an NIH panel, is if you're chronically infected with parasitic worms, it dramatically changes how effective vaccines can be. So you can imagine that in the context of the pandemic, as well as trying to administer other common vaccines. So there's really a tremendous need to develop new therapeutic strategies to treat these infections. And we do know that the immune response and protective immunity to these parasitic infections is driven by type 2 cytokine responses. And so this is really the main area of focus of my lab, and I'll kind of explain how that can loop into kind of disease states that you're going to see in your clinics. So I mentioned that we don't have these parasitic infections in the developed areas of the world, such as the United States and EU. And actually what's happened since then is we've seen epidemic rises in allergies and asthma. So allergies and asthma are driven by that same type of cytokine response, that same type of inflammation, which is the type 2 response. So it looks like we had this whole module of the immune system that was, evolved and originally meant to fight these parasitic worms. We no longer have these pathogens to fight. And now the immune system starts to trigger off on more benign things like food allergens or pollens. And this is a tremendous issue right now. And so it's estimated that about 30 to 40% of the population in the developed countries is suffering from at least one, oftentimes more than one allergic disease. And unfortunately, these statistics are from 2011, and we continuously see the rates rising. And one of the reasons we have trouble treating allergic disease is we really haven't spent much time studying type 2 immunity. It really hasn't been one of our main areas of study in the field of immunology. It's much more common to study antibacterial and antiviral infections, which are the type 1 immune response. So really developing a better understanding of this inflammation serves two pretty important public health concerns, and that's really what our lab focuses on. And that's how I sell it to the NIH. It's difficult to get funding in the United States to study parasitic helminths, but basically the way my lab, the narrative that we bring is if we can really understand what this aspect in this module of the immune system was meant to do, what it's supposed to do, perhaps that can really inform our understanding of what happens when it becomes dysregulated and allow us to understand, or develop new therapeutic strategies to treat allergic inflammation. So that's really how I can get funded in the United States to study parasitic helminth. And you'll kind of see how that all weaves together in my research that I'm going to present today. So what do we actually know about type 2 cytokine responses and the inflammation that drives protective immunity to helminths and allergens? Well, we do know quite a bit. We still have a long way to go, but these responses occur at a barrier surface, an epithelial barrier. This can be the lung, the gut, or the skin. And we know that it's driven by kind of terminally differentiated immune cells. So these are immune cells that are actually present at the barrier, and they're really waiting for something to break through the barrier in order to be able to initiate inflammation and immune response. So those are known, some of the known immune cells are mast cells, obviously your T cells, your B cells, dendritic cells, macrophages, and then a relatively newer identified cell called innate lymphoid cells. And so what happens is upon exposure at a barrier, you have some local cytokines produced by the epithelium, and it kind of mobilizes these terminally differentiated immune cells to activate and initiate the necessary inflammation. And when I was a postdoctoral fellow at Penn, one of the things we had uncovered, in addition to these terminally differentiated immune cells, you also have this hematopoietic program that's initiated, right? So you have all these immune cells that are resident, but you need kind of really a backup force to come and to drive this inflammation. So what we found is what happens is you get these hematopoietic progenitor cells that flux to the tissue. And when they get to the tissue, they then receive signals in the local environment, and then they can develop into other immune cells such as mast cells and basophils and macrophages that help to amplify this cytokine response. And really, when I came to Rutgers around 2015, this was a bit of a new kind of concept of seeing this hematopoietic component of inflammation. And one of the questions we had is this, can we disrupt the development of these progenitors to kind of lessen the inflammatory responses in the context of allergic disease? This became, a nice therapeutic strategy. Perhaps we can interrupt this process that really amplifies the inflammation at the barrier. So one of the first things we did when we were studying these cells is trying to figure out exactly how is it that these progenitor cells that have left and gone to the site of inflammation, how do they differ than the progenitor cells that are in the bone marrow, right? So when I learned about hematopoiesis, you have all the progenitor cells that are in your bone marrow, they differentiate, and then they go to the site of inflammation. And that's actually not what was occurring. What's occurring is there's a subset of these progenitor cells that are going to the site of inflammation and then differentiating at the site. And so there was a basic question here of, have they just switched their anatomical location, or are these cells truly distinct? Are the ones that have gone to the periphery truly distinct than the bone marrow progenitors? And by all of our, kind of surface marker analysis and the things we usually do to phenotype cells, they looked exactly identical to each other. However, if we put them through hematopoietic development assays to see what kind of immune cells they could develop into, one of the things that became really obvious is that these ones that are going to the peripheral sites of inflammation were very good at turning into mast cells. So there was something in their developmental program that had skewed them towards developing into a mast cell. And IL-4 is a hallmark type 2 cytokine that drives allergic responses. It drives IgE class switching. The other thing is, if you look in the culture supernatants of these cells, we saw lots of IL-4. So this made sense to us. The cell is actually not just switching its anatomical location, it's highly specialized to turn into a mast cell that drives the inflammation associated with Helminth infections and allergic disease. And so that was a really exciting finding to us, and suggested, we could look into it in more depth and try to figure out fundamental aspects about things like mast cell development. So just to give you some background on mast cells, this is a cell type we've known about for over a century. It's very unique. If you look at it by just blood smears, you'll see these highly dense granules. So it was discovered by a German scientist, Paul Ehrlich, well over a century ago. We know that it plays important roles in fighting Helminth infections. It responds to antigens derived from Helminths, and it degranulates. These are boatloads of effector molecules, mast cells. They have histamines, leukotrienes, prostaglandins, chemokines, cytokines, numerous effector molecules. And when they degranulate, they derive these strong type 2 responses. So again, they're really important in Helminth infections, and I think to no one's surprise who takes antihistamines every day, they're also critically important in allergic inflammation. They're basically the main driver of allergic inflammation. And one of the ways they operate, and I think people know them best, is they express the Fc epsilon receptor on their surface, which is the high affinity receptor for the IgE antibody. So when you have an antigen-specific IgE, it binds to the mast cell. And then when that mast cell comes in contact, in this case, it's a feline allergen, it'll degranulate and then, again, release all these inflammatory mediators. And that's how allergic inflammation drives. So one of the things we wanted to do is, okay, we have a cell that we know is skewed towards developing into this mast cell. So can we actually investigate that skewed developmental capacity and learn fundamental things about mast cell development? And that's amazingly still an incredible area of need. So although we discovered these cells over a century ago, we know very little about what drives their development. And that's really hindered a lot of our ability to target them therapeutically. This is a very busy slide, but I just show it to kind of give you an idea of, this is decades of data trying to figure out exactly how mast cells develop, like what is their hematopoietic program. So starting from a hematopoietic stem cell all the way down to what's an MCP, this is called a mast cell precursor. And in the literature, all of these various subsets of cells have been said to be a mast cell precursor. And these are all the surface markers that are associated with them. So this is just to give you an idea. There's no consensus on what a mast cell progenitor is. Actually, there's a lot of contention. There's actually a fair amount of arguments between labs about where the mast cell developmental capacity is and what drives mast cell development. So this is a pretty significant area of need, especially given the clinical importance of this cell type. So one of the things we did is we had this, again, cell that we knew was skewed towards developing into mast cells. So we had our regular bone marrow progenitors that didn't have enhanced mast cell capacity, and we had our peripheral bone marrow progenitors. We said, let's just perform some transcriptional profiling on the cells. Let's figure out what genes are associated with this skewed ability to drive into mast cells, to develop into mast cells. And then maybe we can find ways we can interrupt that process. And so this is just a heat map. The red genes are genes that are turned on in the peripheral progenitors compared to the bone marrow resident progenitors. And our thoughts were something within that gene list is going to be a driving factor that allows the cells to become mast cells. And if you just look at the Venn diagrams, we had about 150 genes to work with that were enhanced and were associated with this ability to become a mast cell. And I can't lie, when we were going through the data sets, it was actually really kind of upsetting in many ways. A lot of the things that we saw that were that were enhanced in the cell population that were expressed at high levels were things like mast cell protease 1, mast cell protease 4, granzymes, surface markers, all of these molecules that were more indications that they had started to turn into a mast cell rather than kind of a driving feature of their development, like what's something that's going to push them into that actual developmental program. You know, other things we saw were like RUNX1, which is just a dominant transcription factor of hematopoiesis. So I think many clinicians will know this for its association with leukemias. So really, we were starting to run out of genes that we thought could play a role. And one interesting gene that popped up was this enzyme called carbonic anhydrase 1, or CAR1. And I didn't really know too much about this enzyme family at the time, but we've kind of become experts in the area. So what are carbonic anhydrases? So they're actually a family of basic metabolic enzymes. They're found throughout various forms of life. There are 6 classes. The alpha class is found in mammals. And they're really diverse. So there are 13 family members in this class, and they can be expressed either in the cytosol, they can be membrane bound, they can be secreted, or they can even be mitochondrial in nature. And we don't know too much about them. We know their basic metabolic process that they catalyze, which is basically balancing pH and CO2 homeostasis. So this is a reversible reaction. If you have CO2 and water, you get the production of bicarbonate and hydrogen. So basically, if you have alterations in pH or CO2, these enzymes activate to get things back to homeostasis. But it was really interesting because if it's just a basic metabolic enzyme, why do we have 13 family members, and why are they so uniquely expressed in various compartments? And I can't lie, one of the reasons we decided to look into these enzymes was because we have great inhibitors that can target them. So this is metazolamide, which is kind of a pan inhibitor of carbonic anhydrases, or MZ, as I'll refer to the rest of the talk, but others will know things like acetazolamide. There are few FDA-approved inhibitors, which made it easy for us to kind of study, are these playing a role in mast cell development? So one of the first things we wanted to do is say, okay, is this actually unique to mast cells? Is this expression of this CAR1 enzyme unique to the mast cell lineage? If it's not, then it's not going to be of great, therapeutic target to try to inhibit mast cell development. And surprisingly, when we went through, these are murine immune cells, but these are basophils, mast cells, CD8 T cells, CD4, B cells, macrophages, monocytes. And it was incredible, but the only cell lineage we could find that expressed carbonic anhydrase 1 were the mast cell lineage, which again was very exciting as a therapeutic target to possibly target and interrupt mast cell development. So after investigating the expression patterns of CAR1 in the immune cell compartment, next thing we wanted to test is, can it actually regulate mast cell development? Is it playing an important role in mast cell development? And we have these really nice developmental assays in mice where we can actually take hematopoietic progenitor cells from the bone marrow, culture it with a cocktail of cytokines to drive mast cell development, and then by flow cytometric analysis, we can actually identify mast cells. We basically did this in the presence of either vehicle or the carbonic anhydrase inhibitor metazolamide. So just to orient you here, we do surface marker analysis to identify mast cells. Mast cells express high levels of the c-KIT, or stem cell factor receptor, and then they also express the IgE receptor, FcεRI receptor 1. So basically anything that falls in this box would be considered a mast cell. So if we take our hematopoietic progenitors, we culture them with media, you don't see any mast cells develop. You can see if you give cytokines with the vehicle control, we see a nice population. And then nicely, when we treat it with metazolamide, we virtually eliminated the mast cells, suggesting that, carbonic anhydrase 1 is actually playing a role in mast cell development. The next thing we said, okay, this is, appears to be lineage specific. It appears to be very specific to mast cells. So we did other developmental assays for other immune cells. Specifically, this one is for macrophages. So it's a similar process. You isolate bone marrow, you culture with a cocktail of cytokines to drive macrophage development. Compared to media, you get in our control cultures, a nice population of macrophages, and you can see that's not inhibited when we treat with metazolamide. So we've done a series of these cultures, but again, this is highly mast cell specific, where if you target these carbonic anhydrase 1 enzyme, you can shut off mast cell development. So We didn't at the time have any genetic tools to target the pathway, but anyone working in kind of pharmacologic assays, you know there could always be off-target effects. So it's pretty important to take a genetic approach to target your molecule of interest. And so we had to actually go to the CRISPR-Cas9 editing. We've now since developed all kinds of murine model systems, knockout animals, but at the time we didn't have them. So basically we just took embryonic stem cells and we made guides to target CAR1 to edit it out using CRISPR-Cas9. And you get this CAR1 edited population of cells. It's not perfect. It's about maybe 75% of the cells get edited. And we ran very similar developmental assays, and we looked to see whether or not genetically targeting CAR1 would result in changes in mast cells. And so here you're seeing again c-Kit by, sorry, someone wants to, someone requested to annotate on the screen. I can't seem to get out of it. All right. Can you see my cursor still?
Yes.
Yes.
Okay. I don't know who requested to annotate, but hopefully you won't be drawing on the slides. Sorry, I advanced through some slides here. All right. Sorry about that. Okay. So even with 70% efficiency, we were able to substantially knock down mast cell development by targeting CAR1 genetically. And so here you can see that by percent of the mast cell development from these CAR1 edited stem cells. For the sake of time, I'm not going to show you, but this had no effect on the immune cell development in other compartments like macrophages. So again, this is pharmacologically targeting this enzyme, and genetically targeting it, we can inhibit mast cell development. The next thing we want to know is, can we actually target this in vivo? So culture assays are great, and targeting things in vivo is a whole different story. And we employed one of the parasites we actually study in the lab, which is Trichinella spiralis. So just going to give you a brief overview of this parasite. So Trichinella it causes about 10,000 cases, infections a year. It used to be a substantial problem before we cooked our meats to certain temperatures. But basically what happens is if you ingest undercooked meat, typically pork, the muscle that you ingest is actually encysted with these parasitic larvae. During the digestion process, the acid actually releases them. They infect your intestines. You get these newborn larvae that then encysted in your muscle. And if something consumes that muscle, then the infection continues. But one of the reasons we actually employed Trichinella spiralis is immunity is highly dependent on mast cells and type 2 inflammation. So this is one of the strongest inducers of mast cells that we know about in nature. So if you infect an animal with Trichinella, you will see this massive mastocytosis that occurs that's associated with protective immunity. So if we can inhibit mast cell responses following a Trichinella infection, we basically knew we could inhibit it following any other stimuli. So what we did is we took our animals, we infected them with Trichinella, we treated those animals with either vehicle or methazolamide, and we looked at mast cell responses and whether or not the animals could effectively fight the parasite. So I'm just going to show you snippets of the data. This is a naive small intestine, so this is where the parasitic larvae actually burrow into the intestines, and this is esterase staining here. This is a mast cell, this little red dot, and you can see when we infect with our vehicle, you get this nice mast cell response in the small intestines. And nicely, when we treat with methazolamide, we can see that's significantly inhibited. This is just a section that you can see the difference, and then statistically different. So we knew we could actually, in vivo, by targeting this pathway, reduce infection-induced mast cell responses. And then as we expected, and what's known with Trichinella, when we treated these animals with methazolamide, we inhibit the mast cell response. You see reductions in the hallmark cytokine IL-4. We saw significant reductions in IgE, and most importantly, the mice weren't able to clear the worms anymore. So not only could we interrupt the mast cell response, but then the subsequent type 2 response and inflammation downstream of that was also interrupted. So that was a really important finding for us. Again, this is what we consider kind of a nuclear bomb of mast cells. So if we can inhibit this, we thought we could inhibit almost any form of mast cell-mediated inflammation. And so that's what we started to do. We started to go through other model systems to see whether or not we could inhibit mast cells in the context of, other disease models. Specifically, one we were really interested in was food allergy-like disease. So we have several animal models we run of this in the lab where you can sensitize an animal to ova, which is an egg allergen, and then you can either challenge that animal or mock challenge it. And we did this in the presence of vehicle or methazolamide. And again, we looked for mast cell responses. So this time, food allergy-induced mast cell responses in the intestines. So this isn't as clear to see. Hopefully it can project fairly well on your screens, but this is a challenged animal that wasn't sensitized, so they don't have a food allergy-like response. You can see a couple mast cells at baseline. Again, in our sensitized and challenged animals with vehicle, you can see this nice allergen-induced mast cell response in the intestines. And when we treat with methazolamide, we can virtually eliminate that response. And so other things, associated with this response, the inflammation was also reduced. So infection-induced mast cells we could target, allergen-induced mast cells we could target. And then there are other disease states we were interested that possibly this pathway could help. And I have to really thank the Mastocytosis Society. So there are these, mast cell-related diseases such as mastocytosis and mast cell activation syndrome, where these poor patients just have an overabundance of mast cells developmentally, and they suffer from, really basically chronic or, spontaneous allergic inflammation. And we thought, this pathway could even be, helpful to patients suffering from those diverse diseases. So we have a model of mastocytosis in the lab. We basically take a mouse and we pump it full of growth factors that drive this really strong mastocytosis systemically in the animal. And we did, again, our protocol of either treating with vehicle or methazolamide to see whether or not we could inhibit those mast cell responses. And this, you're just looking at the counts in the intestines, but you can see when we treat with the growth factor, you get this really nice accumulation of mast cells in the small intestines, which is a feature of mastocytosis. And we treat with methazolamide, we can virtually eliminate that response. So at this point, we're, infection-induced, allergen-induced, growth factor-induced. We have been able to successfully target it in models of allergic airway inflammation, also skin inflammation that's mast cell mediated. Virtually every model we've run, we've been able to target it in our animal systems. So now we were at a point of there are really two scenarios that were occurring here, and we needed to get more into the biology of what's happening. So we know when we have either allergic inflammation or parasitic infection at the barrier, we're seeing the egress of some progenitor cells that are going to that site of inflammation and expressing CAR1. And there were really two scenarios that kind of popped into our head. What's happening is the cells don't express CAR1, and then when they get to that site of inflammation, they acquire the expression. And the more exciting scenario was that we actually have a predefined subset of progenitors that are mast cell precursors, and that specific subset actually exits and goes to the site of inflammation. So the idea of having a predefined mast cell subset that we could target was very exciting. But we had to actually determine, was it scenario 1 or scenario 2? So one of the questions we had is, can we identify CAR1 expressing progenitor cells in the bone marrow with a really unbiased kind of discovery-based approach? And the perfect technology to be able to do that is single-cell RNA sequencing. So just to orient everyone, what single-cell RNA sequencing is it basically does transcriptional profiling of each individual immune cell, and then it starts to cluster those immune cells based on their expression profiles of specific genes. So if you express high levels of gene 1, you cluster with other cells that express high levels of gene 1, gene 2 clusters with cells that express gene 2, and then you can see kind of what the defining features of each cell type are and whether or not, they separate from other cells by expressing specific molecules. So obviously we were interested to see, is there a subset that expresses CAR1? Is there a feature of the bone marrow in a naive state where you have a cluster of these cells that express this enzyme? So we took naive bone marrow from mice, we performed single-cell RNA sequencing on them, and this is the way the data is presented to you. These are t-SNE plots. So each of these cells here, or each of these colors, represents clusters of cells that are defined by very specific genes. And not surprisingly, you can find very distinct clusters, 20 of them, if you look at naive bone marrow, and all of them have a small subset of genes that are a defining feature of them. And nicely for us, cluster 13 and cluster 19 were defined by their expression of carbonic anhydrase 1. And so those two cell types, we actually expected one cluster, we found two. So there are actually two predefined clusters in the bone marrow expressing carbonic anhydrase 1. Which again, favors scenario 2 here, that you actually have these cells that are predefined and likely already mast cell precursors that are then exiting to the bone or to the site of inflammation to drive a response, inflammation at the barrier surface. So the next question we had is, okay, this is unfortunately a cytosolic enzyme, right? So this isn't something we can track by binding to its surface. So we need to be able to track these cells and isolate them to functionally define them. So that's what we tried to do, is can we actually track these CAR1 expressing cells from the bone marrow, and can we isolate them and actually look at them with basic assays and developmental assays to get a better understanding of how this pathway is operating in the context of hematopoiesis. So in order to do that, we had to develop a new mouse model, and basically what we did is we just inserted in a fluorescent protein cassette, a green fluorescent protein or GFP cassette. So CAR1 has 8 exons. So what you do is you basically slot in this fluorescent protein after the CAR1 gene, and if you are making CAR1, at the end of transcribing that, you'll make the fluorescent tag, which will allow us to track these cells and be able to work with them and separate them from other progenitor cells. So here's what we have here. These are non-reporter animals. So these are our control animals that don't have the GFP cassette. And we're looking by GFP here. So anything that falls into this circle, into this gate, is considered CAR1 expressing. Compared to our non-reporter animals, when we looked at the bone marrow, we saw a really nice defined population of progenitor cells expressing CAR1. So this was very consistent with what we saw with our single-cell RNA sequencing. And nicely, we knew that mast cell progenitors were also found in the spleen at low levels, and we could actually find a small population of the spleen as well. And so our GFP positive cells are allowing us to separate this progenitor cell from all the other progenitors without really having to permeabilize the cell and get to the enzyme. So this allows for some really nice functional assays that we can run with them. So first thing we want to do, anytime you make a new animal model, you want to make sure it's actually working. Is the GFP actually capturing the CAR1 expression? Is it really linked? So the first thing we did is we took our animals, we took the bone marrow, we sort purified the GFP positive cells, and we ran some expression analysis by real-time PCR. If we take the GFP positive cells, does that actually represent all of the CAR1 in the bone marrow? So when you look at the CAR1 GFP negative cells in the bone marrow, you can see there's virtually no expression of CAR1. If you sort purify the GFP positives, they are in fact the vast majority of CAR1 expressing cells. So that just confirms our animal model is working and that our GFP tag is actually faithfully capturing, the molecule of interest. So now this allowed for really what we wanted to do the whole time. What are these cells and how do they operate? Our thoughts were that they were mast cell precursors, but we had to really prove that by specifically sort purifying them. So basically we took our bone marrow from our GFP reporter animals. This allowed us to separate the CAR1 expressing cells, progenitor cells, from those that are negative. You can put them in this methylcellulose assay. All this is like a cocktail of cytokines that's entirely permissive. So if you put any progenitor cell into it, whatever that progenitor cell can develop into, it'll develop down the pathway. So it's really a permissive culture that allows the cell to go into any developmental direction it's capable of going into. And then after a series of days, you can actually see the cells, these clusters of cells as they develop, and then you can figure out exactly what is the developmental potential of that progenitor. And not surprisingly, considering our other data, this is a media control, so this is without the growth factors acted. You don't see any mast cells when we don't add growth factors to the CAR1 progenitor cell assay. You can see in the positive, the GFP expressing CAR1 cells, the vast majority of the mast cell compartment is within this progenitor cell population. So we saw a high prevalence of mast cells in those cultures. And when we took the CAR1 negative cells, you can see you saw a very small population of mast cells developing. And this is just graphed up here. So basically what this told us is the vast majority of mast cell potential that exists in the bone marrow is in this small population of enzyme expressing cells, which was very exciting. But then the other thing we were looking at was to see, in addition to mast cells, what else can these cells turn into? And this was a bit shocking to us and not something we expected at all. We didn't see any other immune cell lineage develop from these cells, but what we did see, and this is TER119, which is a marker of red blood cells or erythrocytes, we saw that they turned into erythrocytes. And they were highly enriched for erythrocyte development compared to the CAR1 negative population. And this kind of blew our mind at this point. They weren't turning into any other white blood cell, but they turned into mast cells and red blood cells, which was a huge finding, and we were really excited. We thought maybe we had found a branch in the hematopoietic tree that no one else had discovered. And then a week after we discovered this, a paper came out in Nature, but defining the connection between mast cells and erythrocytes. But the first thing we wanted to do was test whether or not if we inhibit CAR, do we actually have an effect on erythrocytes, right? So we were really excited about the therapeutic potential of targeting this enzyme to inhibit mast cells. But if we're going to inhibit erythrocyte development, that's a big problem. And so what we did is we ran some red blood cell culture assays where you take a media to control, you treat with growth factors and vehicle to grow red blood cells. We see red blood cells grow. And nicely, when we treat with methacillimide, we didn't see red blood cell development reduced. So although the progenitors turn into erythrocytes, they don't require CAR1 to be able to undergo that development. So if we target CAR1, we block their mast cell capacity, but we don't affect their red blood cell capacity, which was really exciting. So again, still kind of made this a viable therapeutic target, hopefully with few off-target effects. And as I mentioned, our data kind of suggested that mast cell development is closely associated with erythropoiesis, which was something that was really outside the realm of thinking when you consider hematopoiesis. And again, I thought we had a massive discovery here, and then a week later, this paper came out in Nature, suggesting that erythrocytes are actually highly linked to mast cell development. So this paper was in murine model systems, this wonderful lab out of University of Massachusetts. And basically what they said is that erythrocytes and megakaryocytes were linked to what they call basophils and mast cells, so this BA. So basically, these two cell types, erythrocytes and mast cells, are actually one branch apart. And they had highlighted a series of genes that they thought were responsible for this developmental link, and here they are here. And one gene that's screaming high in that list is CAR1. And that wasn't one of the ones, luckily, that they had thought was one of the, again, it's a metabolic enzyme, so not many people thought it would have this capacity. But it was nice to see that others were seeing this developmental link and that CAR1 was actually associated with kind of where that branch point was. Shortly after that Nature paper came out, in mice, same papers came out suggesting that this pathway is operational in humans. This is single cell RNA sequencing. They put mast cells here right next to erythrocytes. And this lab actually suggested that there is a common erythrocyte mast cell precursor, an EMP, that can either become a mast cell here or an erythrocyte. So they thought by their assays that there is a common progenitor cell that actually branches into either erythrocytes or mast cells. So again, these were nice confirmations that we were kind of on target with what we were studying. We had our bioinformaticists go into their data. And as you start to, this arrow here represents the human progenitor cells. As you start to go towards that cluster that's an erythrocyte mast cell precursor, we asked our bioinformaticist, is human CAR1 expressed? And CA1, which is human CAR1, was highly expressed by that common progenitor that they had identified. So again, this was very much in line. We came at it in very distinct ways, but we were all, very independent labs were coming up with similar findings, that there's a common mast cell erythrocyte precursor, and CAR1 is associated with that cell type. So this was exciting for us. I thought, well, we're seeing this cell population in humans. Can we get leukopaks from human donors? Can we grow mast cells? And what happens if we treat with methacillimide? Can we actually shut off human mast cell development? So we do similar things to our mouse systems, except you use a human cocktail of cytokines and you can grow mast cells. This is actually a wonderful publication, one of the best publications from a protocol standpoint I've ever seen, where you actually tried what they did and it worked beautifully, which is unfortunately not always the case. So here to orient you, these are human mast cells. This is a donor. This is c-KIT positive by Fc epsilon. So again, these are mast cells. In our media cocktail, you don't see any mast cell development. When we treat with cytokines in vehicle, you see beautiful human mast cell development. And we treat with metazolamide, we can essentially shut off that mast cell development. And so we've done this with dozens of donors at this point. It's highly consistent, where we can target this pathway to shut off human mast cell development. So again, in all of our animal model systems and our human culture assays, at least, it looks like this could be a viable therapeutic to target mast cell development. So we're not going to actually get into that. I'll just skip over that. So what this suggested was really exciting to us. So at this point, we have possibly a novel therapeutic upstream target, right? Where we know that this process is happening. We have this CAR1 progenitor stellar stem cell that undergoes development. It becomes a mast cell. And then if that mast cell activates, you get all of the kind of inflammatory responses you'd expect from mast cell activation, the production of leukotrienes and histamines. In the context of allergic disease, this results in itching, burning, mucus production, smooth muscle contraction, and in very severe cases, anaphylaxis. And if you think about our therapeutic targets, they really all target these downstream effects. You're oftentimes trying to target the symptoms that the mast cells are driving, or you're targeting one molecule like antihistamines, right? So mast cells are boatloads of effector molecules. They make all kinds of inflammatory mediators. And histamines play an important role in kind of this allergic inflammation, but it's just one of the factors, which is oftentimes why people that take antihistamines, they feel a little bit better, but they still don't feel great. And so we have a really exciting therapeutic target here to maybe be able to shut off this process and actually prevent the combined effects of all these inflammatory mediators. So every time I present this, people are like, well, why don't we start treating people on mastocytosis, mast cell activation syndrome with metazolamide? Because the dirty secret I'm not showing you is that the amounts that we need to give these animals to get these effects are not ideal for translational consideration. So we had to give animals 2 to 3 mg a day to be able to shut off mast cell development, like in our model of food allergy-like disease. So it was a great proof of principle study, but we're going to cause metabolic acidosis if we're hammering patients with those levels of, someone's requesting again. If we're putting them on those levels of carbonic anhydrase inhibitors. So this is where we really developed the company. We had to get into a whole drug developmental system, like drug development, to try to develop new carbonic anhydrase inhibitors that work better than what we have to be able to take advantage of this therapeutic target. So I never thought I'd get into this, but our lab is now running enzyme assays where we have this whole machine now that we can isolate the CAR enzyme itself. And we can basically test how inhibitors can shut off its ability to metabolize substrate. I won't get into it too much, but basically we have this whole drug development program that's happening over in the Cancer Center, where we're having chemists make these new compounds, and we test them to see how well they inhibit the carbonic anhydrase enzyme itself. This is just kind of what that data looks like. You get this kind of metabolization chart, like this ability of the enzyme to actually break down its substrate. So this is the control where the enzyme, if it's put with its substrate, breaks down the substrate quickly. If we add metazolamide, you can see that process slows down. This is the green line. And some of our new compounds dramatically outcompete metazolamide and actually slow the activity of the enzyme down even better. So we can basically do this at the level of the enzyme first to be able to screen it. Then we have these beautiful assays. These are mast cell developmental assays. These are new compounds that we've developed. This is compound 9. And we can also get a EC50. We can test to see in our culture assays how well and over what concentrations these new inhibitors actually shut down mast cell development. So we can get an IC50 from the enzyme, we can get an EC50 from our culture assays, and then we can also go into our in vivo systems to be able to test these new inhibitors. And we've made substantial progress. This is just a snippet of the data we have. But basically, we now have inhibitors. This is compound 8 that dramatically outcompete methazolamide, work at a much higher concentration. We've now since done all of this work in vivo. So we have compounds that are working in vivo that are not driving metabolic acidosis in the animal models. And this is all funded through the company now with drug development. But we're getting to the point where we think we have some compounds that hopefully soon we can take into patients. We're working closely with the Mastocytosis Society. They have patients that are in dire need of therapies. And so hopefully we can get we can get the safety and efficacy data we need to try to start to get these into patients. But we've made a lot of progress. We're pretty excited about what we've been able to do. So let's see, where am I? So 46 minutes, that's pretty good. All right, I have a lot of people to thank. This was 6 years worth of work from the identification of the enzyme all the way to the drug discovery program. And it's been driven mostly by graduate students, and I absolutely love that they've taken ownership of this project. They've seen it from the identifying the target all the way to trying to get drugs, new compounds into patients. So it's been an exciting process. The first person who worked on this was Everett here, so he's since graduated. Graduated a couple of years ago. At that point, it was taken over by Christine. So Everett's the one who did the identification of the enzyme for mast cells. And then he started to define actually the ability of inhibiting this enzyme to shut off mast cells. Christine did the reporter animal with the GFP. She helped to define all of the mast cell development. Juan did a lot of that work as well. Christina pioneered all of our drug discovery. So she's the one that got that enzyme assay up and running and screened a lot of the compounds. And I really have to thank them. They've been tremendously dedicated to this project. We've interviewed a bunch of patients suffering from mastocytosis and mast cell activation syndrome, and they're highly motivated to try to help those patients. We have great collaborators like the Rivera Lab, the Goss Lab, and the Bilyeu Lab. The core facilities have been amazing here. We're robustly funded by the NIH for the basic research components of that. So basically uncovering the pathway. They don't fund as much in the drug discovery, but nicely through the company, the Venture Capital Group and New Jersey Health Foundation has put over $1 million into our drug discovery program. We really appreciate their support. So after thanking all of them, I just put up this CME again for you, and I can take any questions if you have any.
Yeah, Dr. Siracusa, thanks for that really great presentation, and the translational science of this is just remarkable, and we've taken it really across the entire spectrum all the way through, and so very exciting. We'll look forward, of course, to seeing that there is one of these molecules that you're developing really is able to come into clinical use, maybe in many different ways. So lots of stuff here. I just to go back on just a specific point. You identified two distinct progenitor cells, if I understood you correctly, in the bone marrow, in the group
Yep.
of 13 and 19. And then you also mentioned in the peripheral that you had some in bone marrow, some in the spleen, and some in bone marrow. Was there a difference in these 13, 19 cells? Anything about them?
Yeah, heme proteins. That was the driving feature. Some of them, they look almost identical, but in that single cell RNA-seq, there are a few genes that make them distinct, and it was really heme. And it was shocking to us. I had no idea that mast cells were linked to red blood cells. So, one of the cool things we looked at is if you look at people suffering from mastocytosis and mast cell related diseases, they actually are anemic. And if you go through the literature, it's really remarkable. Most people thought maybe this intense inflammation these patients are suffering from causes the lysis of red blood cells, or somehow inhibits the red blood cells. But now we're starting to think there's a branch point here where perhaps if you're having overabundant mast cell development, you start to lose the ability to become erythrocytes. And so that's why some of these patients are anemic. But those cells are so highly distinct, it's just the heme proteins that separate them. And it's the same in our mouse, and if you go in the human data, very similar. There's a branch point where, for some reason, some of these progenitors start to express the red blood cell associated genes. Other than that, they're extremely similar.
That's really interesting.
The way we think about it now is like when helminths burrow through the barrier, they cause one of their main effects is anemia and bleeding. They cause tons of bleeding because they burrow right through the intestinal barrier. And if you have a progenitor cell that can make mast cells that help to fight the worm, and then also make red blood cells to combat the anemia and maybe help with clotting, that could be evolutionarily a driver, right, of why these two cells are linked, because that's one of the main consequences of a Helminth infection is anemia and bleeding. So, but I did a lot of hand waving there because I just don't know.
No, but it's fascinating. Is there? But if anybody has any questions or comments, please feel free to raise the hand or to just chime in, please, or in the chat box also. That would work as well. In looking at these patients who have mastocytosis, what's driving that? Is there some gene mutation that's doing that, and
yeah,
does that impact the CAR? Is that impacted by CAR?
Yeah. So we're in the process of working with Brigham and Women's right now to figure out, in patients suffering from mastocytosis, do they have an overabundance of these progenitors in the bone marrow? So one of the things they'll do to actually clinically diagnose is they oftentimes will do a bone marrow biopsy. And so we're trying to work on them to figure out, is mastocytosis associated with an overabundance of those cells? But I think importantly, in people suffering from mastocytosis, the majority of them have a mutation in the c-KIT receptor, the stem cell factor. And there are great therapeutics targeting that mutation that are actually working for those patients. The problem is you're targeting the stem cell factor, right? And so patients don't really want to be on that for a long period of time. And we did a lot of work talking to clinicians that are going to employ these kind of these KIT inhibitors to try to handle that mutation. And they'd be interested in having something that they can cycle patients on and off with, to have a couple drugs that they could use. So we're hoping we can help there, but it's nice to see that there is a mutation and there are some therapeutics that are helping to target that mutation. Right now, we don't know how well the CAR1 progenitor tracks with the disease state.
Thank you so much. Question here by blunting, from Juan Bello, by blunting mast-cell-related immune response in the mice infected with trichinella, was the disease natural history affected? Were they able to clear the infection? I think you showed some data on that, but what about that?
Yeah, they're not able to clear as well. So, the mast cell response is really important to try to reduce the parasitic burden in the host. And when we shut that off, obviously that's not beneficial to target mast cells in that context, like it is for the allergic disease. So the animals at that point become highly infected, so substantially more infected. They have a much more difficult time clearing the parasitic worms. Again, that we use that because it's just such a great model of mastocytosis and one of the most potent inducers. So it had all the effects we expected. We shut off the mast cell response, and they had a difficult time clearing the worms. And once we knew we could target that, we thought, we could target anything mast cell related at that point. And that held true for our in vivo model systems.
Yeah, beautiful model. Other comments, questions? One of the things you said early on, and probably was a lead into this drug discovery, I imagine, is that there are many different types, which I had not known of, the carbonic anhydrase inhibitors. And you noted that the one you're developing now is not causing metabolic acidosis, which, of course, clinically, we use the carbonic anhydrase inhibitors to treat metabolic alkalosis in folks with renal issues. So what about that molecule? And are you really differentiating between where your carbonic anhydrase inhibitors are working?
Yeah, so we're doing it in a couple different ways. So there are 13 family members. Our ideal strategy is to target CAR1 because that's our main therapeutic target. So we're designing inhibitors to specifically fit into the groove of CAR1 as opposed to the other, 12 members of the family. So we're trying to be more specific towards CAR1. And then we're also working on trying to kind of slide into that groove more efficiently so we can actually work at a lower dose. So we're trying to be more specific to that one family member, and we're trying to be to have a higher efficiency of inhibiting. So it's a great question about this. There are a bunch of carbonic anhydrase enzymes, and there's a wealth of chemical literature on it, where if you go into it, specific inhibitors target specific family members better. So we actually chose methazolamide because it was one of the ones, when tested against all the family members, it was one of the ones that was much better at hitting CAR1 than the others. And interestingly, we tried acetazolamide, which is also commonly given in clinical situations, and that had no effect. And acetazolamide is not really good at targeting CAR1. It's known to hit other family members. And so luckily our medicinal chemists, again, this is so outside my realm of expertise, it's embarrassing, but they can go into this literature of people who have already looked at all of these diverse inhibitors and see which enzymes they work best against. And it's really a testament to all the literature and all the work that was done prior characterizing these already established, FDA approved inhibitors. That's informed our drug discovery dramatically. And, basic research like that is so critical. I mean, when you have a wealth of research like that to lean back on, I just look at our chemists, they smile and they're like, this is great. I mean, you can, there's so much work done for us as a foundational component. So yeah, we've taken all of that into consideration. It's not my area of expertise by any means, but it's been extremely helpful to see all this other work that's been done on these other inhibitors.
Thank you. That's fascinating. It's really great. Other comments or questions for Dr. Siracusa? Dr. Siracusa, I want to thank you again for really just a wonderful presentation, just really incredible science and that you have done, and that has real application also for human health. And just wish you continued success on all of this, and look forward to hearing more about this in the future. I want to thank everybody who joined this morning, and hope you appreciated this great translational presentation, and just wish everybody a great rest of the day. Thanks again so much.
Yeah, no, thank you very much. It's exciting. I mean, I think the last time presented, we had just identified the target, so it's pretty cool. It was cool for me to put this together and see it all come together. But yeah, when I get these into patients, hopefully it's not too far away. Hopefully I can come back and show you some hopefully beneficial effects of these inhibitors. But it was a pleasure. I really appreciate the opportunity.
Thank you. Thanks very much. Be well.
All right. Take care, everyone.